let's get started. Couple quick things. First, if you still need scantrons, they're over on the side there. Again, alphabetically by last name. Please don't everybody jump up and grab them right now. Second announcement is over on the right. I just noticed that Dr. Lehman in the chemistry department will be offering a course on RNA structure and catalysis. Seems every other time I mention RNA in this class, I say you could have a whole class on RNA. Voila! There is one. So you can take it next term. It's a graduate level course, but if you're really excited about it, I'm sure you could talk to Dr. Lehman and see if you can get that to go. Second announcement is here in the middle. That is that the biology department seminar this week should be really and truly amazing, and I'm really bummed that I'm going to miss it. That's tomorrow at noon by Dr. Michael Vecchione, deep sea diversity. There's just some really amazing things that people have found in the deep sea. I believe he's an ex-curator from the Smithsonian, so he should have some really amazing pictures and, from what I hear, a really nice story as well. Of course, the reason I'm missing it is because hopefully I will be in Florida tomorrow. We'll see. My flight's already been delayed, so maybe I'll still be here on Friday. But nonetheless, there will be a guest lecturer on Friday, Dr. Michael Bartlett, who will talk about transcription. That will not be recorded, at least not by him. You're welcome to record it. I asked him yourselves here, and I will post that on D2L if someone sends me that recording, at least the audio thereof. The second thing about that is he's not going to do clicker questions. Sigh. Okay, any questions about that aspect of the course? Yes, that material will be on the midterm. He will be writing questions for that. Okay, so my apologies. I noticed when I was putting this together that I actually put the wrong lecture number at the beginning of the lecture notes and forgot to change the date. Pretty typical for me. This is lecture 11, and we're going to be talking about recombination, sort of finishing up with that. A little bit on transposition. Again, all of these things are things you could spend a whole term talking about. And then start a little bit on transcription. So the main thing here, this is where we kind of left off last time, is homologous recombination. And really the last step of homologous recombination, I should say the penultimate step, the step before the last step of homologous recombination, generates these things called holiday junctions. So it's a crossing over of strands between your donor strand, this is the one that had the Rec A bound to it, hence strand invasion, and the template strand. So you have these holiday junctions, and in the cases of double-stranded break repair, we've got two continuous chromosomes, and also in meiotic recombination, you have two of these. And people always draw them, well, not always, most of the time, we'll draw these holiday junctions like this, one continuous strand and one crossed over strand. That's not really the way they look. They look a lot more like this. In the lower right here, we have actually crystal structure of a holiday junction with continuous strands on the top, left, right, and bottom. And so a better way to think about these structures, instead of the crossed over like this, is really more in this form. And the reason I'm sort of harping on this is because if you look at a DNA that's like this is clearly symmetrical. So any kind of enzyme that's going to bind to this and do something to it, and when I say do something to it, it's usually going to be your resolution, cutting and religating, it could just as easily cut and religate using these strands, and so you have a left and right, or a top and bottom. There's no way to really tell which one of those is which. Another thing which I forgot to mention last time is once you form one of these structures, there's something called branch migration that can happen. What the heck is branch migration? Basically, what branch migration is once you have one of these crossed over holiday junction structures, they can move back and forth along the DNA, because what happens is each time that, for instance, if this is moving in and then moving out, each time you break a base pair here, the hydrogen bonds, you're forming one right here. And so these crossing over holiday junctions can move a lot relative to each other. And I wanted to take the best way to look at this, as far as I'm concerned, is to take a look at this animation here behind my other plug for classes for next term, the Recombinant DNA Lab, or Advanced Molecular and Cell Biology Research Lab. Close. No. So here, are these going to run? Let's hope they do. Animations looking at, come on. No. Why are we not running the animations here? So, Department of Molecular Biology and Biotechnology, here we go. At Sheffield University, which should be animating these things. Doesn't seem to. Oh, here, down at the bottom. It's maybe a little hard to see. This should look very familiar based on the part of my PowerPoint slides here. So as this DNA here in one part of your holiday junction is unwinding, it's rewinding right here. So breaking and reforming all of the bonds here. And what this means, again, is that these holiday junctions can really move back and forth quite a long way on DNA. And that will bring us back to gene conversion in just a second, because it's one of the main reasons that you end up with gene conversion. So I recommend going to this website. Hopefully you can see the animations of the other bits as well. So what do we mean by gene conversion? Last time, oh, sorry.
I mean, I guess this is kind of on the topic, but last time we were talking about crossovers and how it's the information on the ends is different.
Okay, so basically, would you like me to go over crossovers again a little bit?
Well, I'm just curious, because when you say that the crossover is that the genetic information on the ends of chromosomes aren't the same as what they're initially were, right?
Yeah, so maybe I'll paraphrase your question, and maybe I didn't explain it well last time. So the idea with a crossover is next to one of those holiday junctions, you end up with transferring a whole chromosome or whatever was that copy that you were using onto that same strand. And so you can have crossovers or non-crossovers, and we'll be talking about that a little bit here in terms of this gene conversion. So if it's still unclear afterwards, raise your hand again and ask me about it. So, but the basic idea here is that we had our maternal and paternal chromosomes here, red and blue. Red for the maternal and blue for the paternal. And then after you have meiotic recombination, which is due to this endonuclease that will cut, sometimes you'll have little pieces here, the little vertical blue lines, and that's where when you don't have a crossover that's happening. Or sometimes the majority of the information, in this case of the paternal chromosome, will have exchanged with the maternal chromosome. So that would be your crossover.
So gene conversion is like just a small section, and then crossover is a larger section.
Okay, we'll get to gene conversion in just a second here. So, sorry, no, they're different things, and maybe that's not entirely clear. So the idea with gene conversion is, and I'm getting a little bit ahead of myself here, but bears repeating, is that gene conversion, normally you're going to have two alleles of anything. Right? You've got your paternal allele, maternal allele. Gene conversion can then convert one of those, either maternal or paternal, on the other direction. So you end up with your non-regular distribution of alleles. So your big A, little a, for instance. You'd expect to have 2 and 2 every time when you undergo meiosis. But in this case, you actually end up with 3 and 1. So it's a little different. And how you get there, we'll talk about in just a second here. So, but backing up, so here, any meiotic recombination, which seems to be absolutely required, actually, for getting meiosis to take place, which is interesting and not completely understood why that is. You have to have this kind of crossing over, but also just exchange of DNA wherever you've had that endonuclease which is cut, SPO11 in the case of yeast. Because of that, you'll end up with these heteroduplex DNAs. So what do I mean by heteroduplex? Heteroduplex means that there's one of the strands which has come from, in this case, the paternal chromosome, and the other strand which has come from the maternal chromosome. And this just comes from the double-stranded break repair because you've been chewing in at the ends. That very second step. After you cut, you're going to chew in and make 3' overhangs. So you're always going to have a little bit of information which you've gotten from the other chromosome. So you're going to end up with these heteroduplex structures. It could be a crossing over effect, which is what you have over here on the right, where we have maternal, which completely transfers over to paternal, but still there's going to be this little area in the middle, which is heteroduplex. And one reason that I spent so much time talking about those individual holiday junctions and how they migrate, how far a holiday junction migrates is going to determine how big that heteroduplex is. So lots of migration will have a big heteroduplex as it's moved a really long distance. Less so, you'll have less of that. And so these migration of the holiday junctions will generate these heteroduplexes. Now, just a heteroduplex doesn't mean gene conversion. And so maybe that's getting back to your question, Olivia, about this. And so the fact that you have a heteroduplex just means one's come from paternal and one's come from maternal. If they have different sequences in that maternal and paternal, which they will in some cases, then you could possibly have gene conversion. And so this is what I don't like about this figure. You'll notice I've added possible up here. So this is a possible site of gene conversion, but it's not always going to be a site of gene conversion because the gene conversion really has to be a change of the sequence to have that happen. So if you have heteroduplexes now, and a heteroduplex which has different sequences on either of the two strands, now you'll have mismatches. So that's what's supposed to be shown up here at the top. So now you have a mismatch between dad's sequence and mom's sequence. This is a case of one of those crossing overs, but exactly the same thing could happen if you just have a very small piece. Now we have a mismatch. What happens to mismatches? They get bound to by what? Mismatch repair machinery, which chops out one of the strands and will resynthesize it based on the template on the other strand. So here at the beginning, after you have your double-stranded break repair, your meiotic recombination, you can have a mismatch. But once you've repaired it, now you just have, in this case, the maternal information. So that's what can lead to gene conversion, if those sequences now have been changed to be one versus the other. And that's due to the repair mechanism. It's not due to recombination at all. It's the repair mechanism that happens after you have homologous recombination. Does that make more sense now? Good. Okay, more questions on this? What does that mean? He doesn't have a clicker question quite yet. It's coming. I thought that I had it there. I kind of always remember exactly where I put them. Oh yeah, sorry.
Just a quick clarification.
Yeah.
On the previous slide.
Yeah.
Is the maternal strand always going to be the template strand, or is that just in that scenario?
Okay, so the question here, just to repeat it for people in the back, is it always the maternal strand which gets copied and never the paternal strand? Your female superior? Yes, but not particularly in this case. It seems to be pretty random which one actually ends up getting copied. And so one of the immediate questions that could come up is, say, if you're thinking about mismatch repair, normally when we think about it, it's freshly replicated DNA. So one is already going to have Nicks, or in the case of E. coli, it's already going to be the non-methylated strand. In this case, there doesn't seem to be an easy way to figure out which one is which. So it does seem to be pretty random which one of those copies ends up being the one which is copied. Okay, so again, that's gene conversion. There's another process which in some cases, and I've put it up here in previous lectures, you may have seen it there, maybe see it on some old exams, something called the SOS response, the Save Our Souls response. This is what happens in a cell when stuff is going really badly. And basically, the cell goes, uh-oh, we really need to replicate, but we've got all this DNA damage, but replication's more important than just making sure that everything is repaired first. And this is something that's been extremely well studied in E. coli, again, the SOS response, Save Our Souls response, in multicellular eukaryotes and a lot of smaller eukaryotes as well. Sometimes what happens here is apoptosis, so programmed cell death. But if you're a single cell, undergoing programmed cell death is not such a good idea because you're dead. So one way that E. coli, and again, this has been pretty well studied, deals with this, is we activate Rec-A. Well, what about Rec-A? Where does Rec-A normally do? What did we just talk about Rec-A? Should have a clicker question on Rec-A if nobody's awake on the Rec-A stuff. So Rec-A binds to that single-stranded with the 3' OH at the end for strand invasion. So that's the main thing that Rec-A is doing. When do you get those single strands? When you have lots of double-stranded breaks. So clearly there's a big problem inside the cell. So if you have a lot of Rec-A, then clearly there's a problem with the cell. Turns out that it's not just Rec-A, and Rec-A makes more Rec-A, but there are lots of other genes that get expressed when you have large amounts of DNA damage. And we're not going to talk too much about this, but a lot of the tumor suppressor genes, P53 for instance, is very involved in, there's a big problem with the cell, detected due to a lot of extra DNA damage. So in the case of cells, like eukaryotic cells that are having cell cycle, the cell cycle would be blocked. And then most importantly here is there are these excuse me, things called error-prone DNA polymerases or translesion DNA polymerases. So a couple of people asked me in office hours, now you've talked about DNA polymerase 1, DNA polymerase 3, DNA polymerase alpha, delta, epsilon. What about all the other polymerases? Well, many of those other polymerases are these kinds of polymerases, which are basically polymerases which are your last ditch. Now, you need to replicate your DNA, but there's this DNA damage which has happened that you somehow need to get past. How that happens, the normal replicative DNA polymerase, this would be DNA polymerase 3 in bacteria, Pol delta epsilon in eukaryotes, will hit one of these areas of DNA damage, and instead of the repair machinery coming in, what happens is the sliding clamp will bind to one of these translesion polymerases or error-prone polymerases that just puts it in whatever nucleotides it can find. So they're very low fidelity, i.e. error-prone polymerases, just to get past that DNA damage. This DNA damage can get repaired later on, but you've got to finish replication. So that's the, that's the process here. The main protein in eukaryotes that blocks a cell cycle is this protein called ATM, which I always mispronounce, so usually goes by ATM. That's another detection of these DNA damages that says, okay, we got real problems. We're either going to, again, particularly in eukaryotic cells, undergo apoptosis or express some of these translesion polymerases. Another way of looking at that is here. This is the case in E. coli. You have your sliding clamp, again, which is beta. DNA polymerase 3 hits one of these pyrimidine dimers, and the replicative polymerase falls off. One of these error-prone polymerases, Pol 4 or Pol 5. Again, we already had 1 and 3. Now these are 4 and 5, which will replicate across from this DNA damage. In one case, it actually puts in the correct nucleotide. In the other case, it doesn't. And this seems to be pretty random. Then this polymerase will fall off. The replicative polymerase will come back on and continue to replicate. What do you notice about this particular polymerase versus this one, other than the color? Very similar shape, but what else is different? Sliding clamp. No sliding clamp associated with these polymerases. Why? It's not because of dimers, mismatch repair. What do you want to make sure you don't do if you have one of these polymerases that can put in nucleotides willy-nilly? You don't want it to be processive. You don't want it to stay on the DNA and make a whole bunch of mistakes. Where does processivity come from? Sliding clamp. So doesn't associate with the sliding clamp, therefore it doesn't put in a whole bunch of these incorrect nucleotides. Okay, so that's it for DNA repair. Again, whole course could be taught on DNA repair. So today we mostly talked about double-stranded break repair. Last time we talked about this non-homologous end joining, and today homologous recombination, and then this translation synthesis, which happens when all of the bets are off and hell or high water, just replicate through and finish replication happening. More questions on this? Yeah.
So what is it just a different polymerase on one slide that is showing it connecting with the clamp compared to the one that it drops the clamp completely? Because the first slide where it's got the clamp on there, that one,
Yeah.
staying on. Does it depend upon the mutation?
Yeah, so the question here has to do with the difference between these two different slides, one of which is from our textbook and the other one is from a different textbook. The case here, if I remember correctly, is it's a different one of those translesion polymerases. So there are multiple different translesion polymerases, and some are going to be helped out by the sliding clamp, and they've got these extra little Pac-Man structures up here, which will help bring in some of these polymerases, but they do not associate. They're non-processive, and the important thing is these are non-processive, because they can't be processive. If they were, you're going to be introducing all of these crazy changes. Okay. Other questions on DNA repair? Switch gears and talk about transposition, jumping genes. So there are, come on, if we can get this to work, that would be nice. Transposons. Jumping genes, why Barbara McClintock, probably one of the most intuitive molecular biology and certainly genetics researchers in the last century or so discovered these things. Nobody believed her for years and years and years, and then eventually ended up getting the Nobel Prize because they finally figured out that she was right and everyone else was wrong. But one of the very few people to actually have a non-shared Nobel Prize in medicine. So she discovered these things, the jumping genes or transposons. We now know, turns out she was mostly looking at this kind of transposon up here. There are really three major kinds of transposons that are known, literally jumping genes. They differ in a number of different ways, and these are sort of the important ones, and we'll see why this is in just a second here. They're the so-called DNA-only transposons, so it's literally a piece of DNA that jumps around. If you just look at a genome sequence, how do you know? It's because these guys have short inverted repeat sequences at either end of their transposon. And we'll look at inverted repeats and direct repeats on the next slide. On the other hand, what we have in our genome are lots of these retroviral-like retrotransposons, which have directly repeated sequences at either end of their genome. And then, as we'll see in just a second, our genome is absolutely loaded with these so-called non-retroviral retrotransposons. Why do we call them retrotransposons? They're retrotransposons because they have an RNA intermediate. So all of these, the retroviral-like and the non-retroviral-like retrotransposons, make an RNA copy, and that RNA copy then gets put into DNA to jump into the rest of the genome. So the DNA transposons, most of them anyway, there's some exceptions, are just these cut and paste. So it takes a piece of DNA, takes it from one place in the genome, and puts it somewhere else in the genome. The retrotransposons, you make an RNA copy, and that RNA copy then goes and gets inserted somewhere else in the genome. What that means is these multiply, because you don't get rid of that first one. You haven't moved a gene from one place to another. You've made a copy and put that copy somewhere else in the genome. So again, RNA intermediates that are here. This is just DNA. There are lots of different ones here. We're not going to talk about the details here. So what are direct and inverted repeats? So a direct and inverted repeat, and I, again, like to draw these up, makes it much easier to think about. A direct repeat just means you have the same sequence of nucleotides from 5' to 3' on the same strand. So for instance here, GAA, TGC, CA, GAA, TGC, CA, T in this case. These N's here in the middle just represent any nucleotides. These should be any of the 4 nucleotides here. These could be further apart, long distances, short distances, etc. Anytime you've got the same sequence on one strand, that's going to be a direct repeat. Okay, I'm emphasizing on the one strand. If you now have the same sequence on an opposite strand, that's going to be an inverted repeat. So again, I have the same sequence here, GAA, TGC, CAT. Now flip it on its head from 5' to 3', GAA, TGC, CAT. This is your inverted repeat. So direct repeats, inverted repeats. And if you look at some of the old exams, you'll notice I love to ask questions about direct repeats and inverted repeats. So if for some reason you're not clear on these, come talk to me, give us some examples, etc. So why do we care about these transposons? I've probably beaten to death right now the fact that minimal amounts, about 1.5% of the genome, is protein coding. Almost half is transposons. This is the human genome. So very, very large numbers of transposons are present in our genome. They're actually relatively small numbers of DNA-only transposons, but they're still more than the protein coding sequences that we have. Many more of them are these retroviral-like retrotransposons, and then the vast majority, over 30% of our genome, is these non-retroviral-like retrotransposons. And that's probably because of what I mentioned before. These get amplified. You're making a copy and moving a copy. And literally over billions of years of evolution, these have replicated and ended up in many, many copies in our genome. So let's take a look at how some of these things work. Bacterial transposons. Who cares about bacterial transposons? Well, if you're a patient or an MD, you should care a lot about bacterial transposons. Why is that? Because many of these bacterial transposons contain antibiotic resistance genes. So a lot of the highly antibiotic resistant bacteria that are causing all kinds of problems, some of the multi-drug resistant bacteria, the superbugs, are carrying these transposons which give antibiotic resistance to the bacteria that's picked them up. So if you look at these, they've got all have the same structure. Again, some of these have these antibiotic resistance genes, some of them don't. They've got the direct repeats at either end. That's what these little red boxes are supposed to be. And then also have a transposase gene that's between these direct repeat sequences. And this piece of DNA is autonomous by itself. It can literally, if you just make the protein that's coded by that transposase gene, it can chop that piece of DNA out and move it to somewhere else in the genome. How does this work? We have this transposon in the DNA, the transposase, which is again is being made by the gene, which is coded in this transposon, binds to the ends of this transposon. So when it binds here, there are these inverted repeats that it binds to. Okay, inverted repeat, direct repeat, what's going on here? Well, these are dimers. So 2 copies, a homodimer of the same protein, but they bind back to back. And what that means is whatever they're going to bind to is going to be inversely oriented. That inverse orientation you see with inverted repeats. So those are in the transposon sequence here. Once it's bound to these, then this transposase cuts the transposon out of the DNA. Now it can go and find a new place to jump into. The transposase, again, also will cut here and insert the transposon. One thing you'll notice are these little red arrows here. You'll notice these red arrows are not lined up with each other vertically. They're offset a little bit. this offset means that you're going to have a gap in your DNA after you have the transposon that's inserted. And it's hard to see here, but there's a little black segment right here. There's a black segment there and a black segment right here. And that's because of this offset. These two pieces get pulled apart, and there'll be a little piece of template, a 3' OH. What happens if you have a template and a 3' OH? DNA polymerase comes in, it will fill that in. But because these sequences right here are complementary to each other, these black sequences right here are now going to generate a directly repeated sequence outside of your transposon. So just to repeat here, we've got inverted repeat sequences that the transposase binds to, direct repeats which are generated by this offset cutting, and then the DNA polymerase filling them in. So if you happen to have a genome sequence that you're staring at, and you're cruising along and you see, oh, here's a gene that looks like a transposase. Okay, now we've got some inverted repeats on either side of that gene, and then direct repeats outside of that. This is really good indication that this is a DNA transposon, DNA-only transposon that's been jumping around in your genome. So just literally by looking at the sequence, you can say, oh, direct repeat, inverted repeat, transposase, inverted repeat, direct repeat. That's going to be a DNA-only transposon. What does this look like? Here we have the dimer of the transposase bound to this particular DNA. And again, this is basically the only main thing to show here is a dimer. These guys are binding in back to back or front to front, depends on how you want to talk about it. This sequence up here is going to be the same as this sequence down here, but it's flipped over because the two parts of the transposase are flipped relative to each other. So that's the DNA-only transposons. Again, big problem as far as antibiotic resistance in bacteria. Our genome doesn't have very many of these, but it's got way more of these other kinds of sequences, which are viruses, and so they're totally cool, which is another class you can take next term. So retrovirus replication, we spent a whole lecture talking about this, so if you want to know more details, take my class next term. Retroviruses, amazingly interesting organisms. Yes, I call them organisms, where you have production of an RNA molecule. And that RNA molecule has reverse transcription that takes place. Where did my pointer disappear to here? Try again. Pointer, there we go. Oh, no, it's showing up here, not on this screen. Oh, well. So retrovirus replication will take place where you release an RNA, which is in the middle here, going with the arrow coming up. That RNA is made into DNA. That process is the reverse transcriptase, the thing that Crick didn't think was happening in the central dogma, although later he said, oh, of course, it was going to be happening anyway. So RNA goes to DNA. That DNA then gets inserted into the chromosome, which is that big orange piece up at the top there. Then once that has been integrated into the genome, that then can be copied into RNA. The blue lines, those blue lines then can go back and make more copies of DNA, get inserted in the genome, and so on and so forth. In the case of a retrovirus, these RNAs actually get packaged into infectious virus particles, which will go off and infect another cell and go through this whole process again. But that mechanism of going from RNA to DNA and then popping into the genome is absolutely identical for these retroviral-like retrotransposons as the viral replication process. What does this? It's the reverse transcriptase. This is the RNA-dependent DNA polymerase. Again, it takes an RNA copy and makes actually a double-stranded DNA out of that single-stranded RNA copy. And all of you notice here immediately that it looks like a DNA polymerase because it's got its hand, thumb, fingers structure, right? All of you noticed? Maybe not. But it's got a very similar structure to the DNA polymerases. Doesn't have a proofreading activity, but has a very similar kind of activity and also a very similar kind of structure. Once that double-stranded DNA has been made, that process actually is going to give you a directly repeated sequence that's inside the virus genome. How that happens, take virology next term, we'll talk about it ad nauseum. But it generates these long direct repeats. These long direct repeats are actually right here outside of these arrows here. So this viral DNA is cut, binds to the integrase, which has a different sequence and a different structure to the transposase, but the activity is practically identical. That will now bind to some other place in the genome. Again, offset cuts, giving a little gap in your DNA. That gets filled by your repair machinery. But now, instead of having inverted repeats, we now have direct repeats in our viral genome or our retroviral-like retrotransposon. So now we have a short direct repeat that's generated due to this offset cutting, but then the long direct repeat, also known as the long terminal repeat in viral genomes, that's at the end of each of these transposons. A lot of people also talk about these transposons being LTR transposons, long terminal repeat transposons, because they don't like the viral terminology for them. I don't know. I quite like it. The last kind of retrotransposons that I wanted to talk about are these non-retroviral retrotransposons. They're much less well understood how they function, and that's partly why I don't have a whole bunch of stuff written down here, because the details are unclear. But the basic message here is you have a RNA with a poly-A tail that's been added at the end. And yes, we'll talk about how poly-A tails happen later on. Hopefully, poly-A tails mean something to you in terms of messenger RNAs. Eukaryotic messenger RNAs have these poly-A tails. So the poly-A tail, then at the end of this RNA of the non-retroviral retrotransposon, this binds to the protein that's encoded in this particular RNA. This protein is a reverse transcriptase and endonuclease. So what does endonuclease mean? It means it cuts in the middle of your DNA, and it's a reverse transcriptase, takes RNA, makes it into DNA. So what happens here, you have the protein, binds to this poly-A tail, cuts the target DNA, and now reverse transcription is going to copy this RNA. What's present at the 3' end of this RNA? A poly-A tail. So what are you going to see? Stretch of T's. So A's and T's are what you're going to see at the end of all of these non-retroviral retrotransposons. And these, the line elements, are also called so line element is long interspersed nuclear element. Horrible name. But it's one of the most common repeated sequences that we see in our genome. Between, depends on how you look at it, 10% up to 20% in some cases. This then holds a multi-step pathway producing the second DNA strand. That's what we're ignoring all the details here. But what happens, again, you've got one of these copies which has been made in your genome with ATs right at the end of it. And our genome is absolutely packed with these things. There's a difference between the lines and the signs. What's the difference between a line and a sign? The first letter. So large interspersed nuclear elements and small interspersed nuclear elements. What's the difference between large and small? Either you code this protein right here, the reverse transcriptase endonuclease, in this RNA, or you're lazy, or this particular gene has been chopped out of that particular RNA, but you still have all the signals so that this protein can copy the RNA that doesn't encode that. So the short interspersed nuclear sequences don't encode the reverse transcriptase endonucleases, and the long interspersed nuclear elements do. And those signs seem to be parasitizing on those long elements to make many copies of those smaller elements. And between those two, you're talking almost 35% of our genome. Yeah?
No lines means no signs then, because they can't produce the reverse transcription.
Right. So the question here, again, just to repeat it, would mean if you don't have a line, then you're not going to have signs. Well, at least not going to have signs which are jumping around in the genome. And one of the immediate things you're probably thinking, or I hope you're thinking, is how the heck can we have all these genes jumping around all the time and not just be completely messed up? So it turns out that the vast majority of these elements in the genomes are disrupted in some way. So most of those lines, most of those reverse transcription endonucleases are not active. So there are very few of them, probably through selection processes. There are a few which are active. And so there are few of these things that jump around. But for the majority of our genome, these seem to be dead retrotransposons. Okay. More questions on these reverse transcription. I wanted to briefly talk about site-specific recombination, partly because it's viruses, and viruses are totally cool. But also, this turns out to be a really useful technique that molecular biologists have now used and molecular geneticists to make particular mutations in genomes. So site-specific recombination, unlike your transposases and your retroviral and the non-retroviral reverse transposition, these are specific. They go into one specific sequence in your genome. So the way that these work is there's a specific sequence in whatever it is that's going to recombine and the genome itself. This case is what you often see with viruses. We'll talk about that in just a second. But you can also have, if you have these two inverted repeat sequences in the genome, if these guys can now line up with each other, just like they've done here, instead of having integration, and then when you reverse this, you end up taking this piece of DNA out, this piece of DNA can be flipped back and forth. And it turns out that flipping DNAs back and forth is really useful. It turns out there's some bacterial pathogens that use this mechanism to express different genes so they can escape the immune system. And all that this is are sequences that are either directly repeated, so that's why I'm beating on the direct and inverted repeat sequences. Directly repeated sequences you have up here, this will be either integration or excision through the activity of these enzymes. If they're inverted repeat sequences, you end up just flipping the piece of DNA that's between the two. The best understood of these site-specific integrations is something called the lambda integrase. Lambda is a bacterial virus, has these repeated sequences in the viral genome and the host genome. There's an integrase protein, which now is a specific recombinase that will cut this DNA and insert it into the genome. And this same integrase can bind to this DNA and chop the virus genome out. Again, we'll talk much more about this in virology next term. There are, this is that process here. The lambda genome is released inside the cell. It circularizes. This now can integrate into the host genome. Cell division happens. Then you have an induction event. What the heck is an induction event? An induction event turns out to be a real problem for the cell. The SOS response. Talked about the SOS response, huge amounts of DNA damage that cause these error-prone polymerases, etc. Well, if you're a virus sitting in a genome and you don't want your DNA to get messed up, what happens when you have an SOS response? You say, it's time to get the heck out of Dodge. And so what happens, and again, totally over-anthropomorphizing here, is the virus genome is then circularized, it gets out, makes more virus, and goes back and does the whole process again. And again, we'll spend a whole lecture talking about this next term. Why do we talk about that in this class? Yeah, who cares about bacterial viruses? It turns out that understanding bacterial viruses has turned out to allow us to do some really amazing genetic techniques in lots of different cells, and particularly in eukaryotic cells, because they don't normally have these DNA sequences. So if we use recombinant DNA techniques to put in these extra sequences into these cells, we can tweak them. And when I say tweaking, mostly that has to do with doing a deletion of a particular gene. So a lot of time people will talk about these genes which are phloxed. What the heck is phlox? So phlox just means flanking LOX-P sites. Okay, well, what the heck is a LOX-P site? LOX-P site, excuse me. The LOX-P site is just a directly repeated sequence that a recombinase, and in this case it's very much like an integrase, can bind to and chop out this piece of DNA. So using recombinant DNA techniques, you can put whatever, and here it says gene of interest, I always like to call this your favorite gene, YFG, into a particular cell that you want to delete at some time. And the way that you get that to delete is you put in this gene which is going to make the recombinase. The cell normally doesn't have this at all. It's only when you put that gene into that particular cell, now you can get the gene that you phloxed to be removed. And so this way you can learn all kinds of things about the activities of particular genes in particular cells. So it's a ridiculously powerful technique for looking at gene regulation and gene deletion. Now people are doing CRISPR all the time. Who cares about all of this stuff? But we'll hear about CRISPR later on in the course. So what about transposons? We talked about the DNA-only transposons. Those are important because they're often carrying antibiotic resistance genes, and literally transposase that binds to inverted repeats at either end and goes and pops it into the genome somewhere else. Retroviruses have these direct repeats at the ends of the viral genome that gets moved around, but still through a viral integrase. The other retrotransposons are going to end up with AT stretches, and again, the lines, that line protein is the main thing which is leading to those jumping around, and signs get moved around by that line 1 protein. And then site-specific recombination, lambda. Oh, this process is also called recombineering, which is using recombination in a genetically modified cell to do this kind of gene deletion. And somehow I seem to have deleted all the clicker questions. Not quite sure how that happened. Oh well, drat. So again, hopefully this is all review here. Want to just get started briefly in the last 10 minutes, 15 minutes or so with transcription. So before we do that, more questions on transposons. Okay, transcription. This is exactly the same image that I used back in lecture 1. So DNA replication, more copies of DNA. Transcription is making an RNA copy from this DNA copy in the first place. So what does that? It's the DNA-dependent RNA polymerase. And here we're pretty much always, when I say RNA polymerase, it's going to be a DNA-dependent RNA polymerase. And what I mean by DNA-dependent, it's using a DNA template to make RNA from. Today we'll talk a little bit about bacterial transcriptional initiation, and I'll let Dr. Bartlett talk about eukaryotic and archaeal transcriptional regulation later. We'll talk about translation well after, probably getting towards the next midterm. So why transcription? Why are we so excited about transcription? One of the things that's really important about transcription, just like you see with these retrotransposons, is it's an amplification process. You can have one piece of DNA and make many, many copies of RNA from it. Now, as I've also mentioned, there's only this tiny little part, maybe 1.5% of the genome, which is actually protein coding. So how do you get all of these protein coding sequences, which are these vertical gray lines here, made into protein versus all of this other junk which is present in here? And how do you figure out when you get a genome sequence, which again we'll talk about particularly at the end of the class? Really easy to get genome sequences. Way harder to figure out what's being made into protein and what's being made into RNAs. So there are a couple of ways of figuring that out. One of those is what's shown here in the blue. And in the blue is just how frequently you find that sequence or a similar sequence in all kinds of different organisms. And so this is conservation. Again, getting way back to lecture 1. Conserved sequences are probably going to be important for function. And if you look where these genes are, you'll notice that the blue lines at the bottom are all high wherever you have each of these vertical gray lines. You also see some areas where you've got conserved sequences that there's no obvious protein conserved sequence, and we think these are probably important sequences for either regulatory reasons or potentially non-coding RNA sequences which are there. We'll talk a little bit about splicing later on. This is how you go from one RNA to multiple different RNAs, and this overall process. Another thing to look at here, so the bottom is conservation. This blue stuff is conservation in lots and lots of different organisms. The triangles here have to do with differences between some of our nearest relatives, particularly in this case the Neanderthals. There's been great progress in the last probably 5 to 10 years sequencing what's called ancient DNA, and now we can actually compare Neanderthal DNA to modern human DNA. And there's actually quite a lot of Neanderthal DNA in modern human DNA as well. Interesting story that's, again, way beyond this course. But if you're interested in things that make us modern humans different from Neanderthals, you might want to really focus in on some of the sequences which are different. So again, getting back to conservation. If it's conserved everywhere, it's probably going to be important for lots and lots of different things. If it's not conserved, that might tell you something about the differences between different organisms. And one of the things they point out here is this cluster right here. Lots of different sequences in Neanderthals relative to modern humans. So maybe that's something about differences between modern humans and Neanderthals. What are we making? RNA, again, this is exactly the same slide as I showed before. What's the difference between RNA and DNA? Basically, 2 big differences as far as I'm concerned. The book gets way into the single-stranded stuff. I don't like that. But the 2 big differences as far as I'm concerned, it's ribose versus deoxyribose and uracil instead of thymidine. So, again, quote-unquote single-stranded. Why do I say quote-unquote single-stranded? Because most RNA looks like this. That doesn't look like a single strand to me. It's double-stranded. It's bound very often by base pairs to other pieces which are present in here. So that's your RNA. How is RNA put together? RNA is polymerized through the activity of this DNA-dependent RNA polymerase, practically identically to how we talked about DNA polymerization. So you have a 3' OH. That 3' OH is going to attach to a 5' phosphate of a new nucleotide coming in. Again, hydrolysis of those pyrophosphate. The pyrophosphatase is going to break those down. So in terms of the actual chemistry of these particular reactions, practically identical. Uracil is different, and you have this extra OH here. Turns out this extra OH is really important for a lot of the catalytic properties that RNAs have. And take Dr. Lehman's course next term to hear all about how important those different things are here. What kinds of RNA do we have in the cell? Oh, sorry.
So I just had a question about the non-coding RNAs. So
Yeah.
I was thinking like they maybe didn't code for proteins, but so maybe they're the catalytic RNAs or something. But what exactly are non-coding RNAs?
Okay, so perfect lead-in actually to this slide. Thank you. So what are the non-coding RNAs? So non-coding RNAs, by definition, are actually non-protein coding DNAs. You're exactly right. And what are all of the other ones? And the answer is we don't know what all of the other ones are. We know a few of what they are. And the most important of those are these ribosomal RNAs. So the ribosome is a so these are non-coding RNAs. They're not coding for protein, but they're critically important in terms of getting translation to happen. So the vast majority, actually, of the RNA in any cell are these ribosomal RNAs. Transfer RNAs that we mentioned as well, also very important for translation, also non-protein coding RNAs. So these are the ones that we understand reasonably well. The coding RNAs are the messenger RNAs. 3 to 5% of the RNAs in the cell are these, so pretty small number, actually, relative to the ribosomal RNAs and the tRNAs. And then you've got this whole list of non-coding RNAs here as well, above and beyond the tRNAs and the ribosomal RNAs. We'll talk about the small nuclear RNAs. These are very important for splicing. We'll talk about SNO RNAs. These are important for modification, particularly of the ribosomal RNAs and the tRNAs. And then a bunch of other RNAs, particularly these microRNAs, small interfering RNAs, and Pee-wee interacting RNAs. These are the RNAs that people threw away all the time because they were down at the bottom of their gels, tiny little RNAs, contamination, blah, who cares? Turns out they're absolutely critical for regulation, also for regulating transposons and transposase activities, lots of things that are going on with these small RNAs. And that's been, huge area of research recently. And then there's also these so-called long non-coding RNAs. Now, long non-coding RNA, what do you mean? Basically, it's just more than about 100 nucleotides, and we're not really sure what it does for the vast majority of these things. We'll talk a little bit about some of them later. They seem to be important for coordination purposes. But as I mentioned way back when, and probably kind of sort of beating the dead horse on this as well, is that we don't know what huge numbers of these RNAs do, and we're still trying to figure out quite what's going on there. As I mentioned before, we talked about number of protein coding genes in the human genome, about 20,000 of them. Non-coding RNAs, a bunch. Definitely more than 10,000 and probably way more than that as well. So we don't really understand quite what's going on there. Yeah, Olivia. So exactly. So the mRNAs are the only coding RNAs. And so when we talk about non-coding RNAs, it's really the vast majority of RNAs are actually the non-coding RNAs, even though we mostly are going to think about the messenger RNAs, which may be a myopic view just because of the way we've been thinking about a lot of these things. So hopefully this is a review. Again, transcription, you can make multiple copies of one piece of DNA, or you can also make relatively small number of copies of RNA from a particular gene. And this we'll talk about after the next midterm. This is all about regulation. How much of a particular RNA you get is going to be dependent on all kinds of other things, including the chromatin structure, which we've already talked about. So as far as we're concerned, at least for the next couple of lectures, we're going to talk about just how you make the RNA in the first place, not how much of that RNA you end up getting. Again, that's for later on. What does this process of making the RNA? Mentioned this multiple times already. It's the DNA-dependent RNA polymerase. What does it do? It makes a complement of one of the strands of your DNA, and just by definition, the template strand is the one which is getting copied. Also, always made 5' to 3', again, not surprisingly, because this is exactly the same mechanism as you see for DNA polymerases also, which all go from 5' to 3'. Big difference between DNA polymerases and DNA-dependent RNA polymerases. They don't need primers. So you need a template, but you don't need a primer. And this is why the DNA primase is actually an RNA polymerase. It doesn't need a primer. What was one of the big problems with DNA primase? Low fidelity. Turns out that DNA-dependent RNA polymerases are also low fidelity. They make mistakes, but these mistakes don't get continued in the germline. They're not something which is passed along genetically unless, of course, you have a reverse transcriptase, which is putting it back into the genome. So you can basically get away with a lower fidelity polymerase than you can with a DNA polymerase. There's a little bit of quote-unquote proofreading that happens here, but for the most part, it's just polymerization which is happening through your genome. This RNA, once it's made, comes out of the RNA polymerase and is relatively rapidly released. So if you think about how you're getting the transcription happening here, if you think about what happens with your DNA polymerase, just a second, you have the two strands which are pulled apart by the helicase, and then your single-stranded DNA binding protein, and so you end up, again, with these highly processive enzymes which are making these extra strands. There's no helicase activity per se here pulling apart those two strands. There's just a transient pulling apart, so temporary pulling apart of the two strands up to about 10 nucleotides, which is where that RNA polymerase is making the RNA. So yeah, your question. So the question is, if the RNA polymerase is low fidelity, won't it affect your protein? The answer is definitely yes, but these are things which are not being passed along genetically. And we'll talk about some of the mechanisms so that the cell knows that there's been a problem with the RNA or a problem with the protein, and we'll talk about that later. But there are control mechanisms to deal with that. Also, when we talk about the genetic code, there's lots of flexibility in the genetic code. So you can actually end up with the right protein even if you have the wrong, quote-unquote, RNA sequence, which is there. But yes, you certainly can. It just seems that since it's not being passed along genetically, that it's not as big a problem. The other thing, which of course all of you have noticed, is that the structure here doesn't look like a hand. It's a very different structure, which is actually very interesting evolutionarily speaking. As I showed before, the reverse transcriptase, that looks like a DNA polymerase in terms of its structure. RNA polymerases have these very different structures. And why that is, even though they have practically identical enzymatic activity, is a really open question and may tell us something about how RNA evolved or DNA evolved, etc. Through through the evolution of life as we know it. One other last thing to mention down here, I've talked about amplification of any given gene. This is an example down here at the bottom where you are literally amplifying one gene into a whole bunch of RNAs. So this is one gene down at the bottom here. Here are all the RNAs coming off of that one particular gene. So clearly being heavily amplified here. So I want to finish up just going through this bacterial transcription process. Turns out the same thing is true in eukaryotic and archaeal transcription as well. It's a whole process where you have the polymerase coming down and binding onto the DNA where it's going to start, then undergoing a number of changes in structure or conformational changes which lead to production of this RNA and eventually ending the RNA later on. So let's look at this. Where do RNA polymerases bind to the DNA and start? What's the equivalent to an origin of replication? It's the promoter. So the promoter is where the RNA polymerase is going to start transcribing. In bacteria, how does it know where to find this? It's through this protein called the sigma factor. We're going to talk a lot about factors throughout the next couple of lectures. Factor is just another name for protein. So the sigma factor is a protein that helps the polymerase bind to the promoter. After it's bound to the promoter, there's this thing called a closed complex. What the heck is a closed complex? Closed and open here just means, are the two strands together or have the two strands come apart? So you form a closed complex when the two strands are together, then an open complex when these two strands come apart. There's no separate helicase here. It's the RNA polymerase itself that pulls those two strands apart. So again, a big difference relative to what we see in DNA polymerization. Then there's what we call abortive initiation. This pumps out short RNAs. In the book, they talk about scrunching. The RNA polymerase sits there and plugs out a few nucleotides, and then eventually moves from this abortive initiation to elongation. Why does this seem to be so important? The main thing that seems to be important here is that these DNA-dependent RNA polymerases are incredibly processive just by themselves. They don't need a processivity factor. Just the RNA polymer itself is incredibly processive. Once it gets past this abortive initiation, it will transcribe. And some genes in the human genome are literally hundreds of thousands of nucleotides long. So once you make the decision, again, to over-anthropomorphize, you really want to be sure that that's a gene you want to transcribe. And then you get to the end, that's termination, and I will let Dr. Bartlett tell you all about how these things work on Friday.